For 2,000 years, the Jewish people were scattered across the world, from Jerusalem to Rome to Spain to Germany to Poland to Morocco to Iraq to Ethiopia to India. They lived in different countries. They spoke different languages. They ate different food. They looked different from each other. An Ashkenazi Jew from Poland and a Mizrahi Jew from Yemen share a religion and a sacred text. Looking at them, you would not assume they share anything else. The question that geneticists asked was simple. After 2,000 years of separation, living among different populations on different continents, are Jewish communities still genetically related to each other? Or have they become genetically indistinguishable from the populations they lived among? The answer shocked everyone, not because it went one way or the other, because it went both ways simultaneously. Jewish populations across the world, from Krakow to Casablanca to Baghdad to Addis Ababa, share a genetic core that traces back to the ancient Levant, roughly 2,000 to 3,000 years ago. 2 millennia of diaspora, of living among Europeans, Arabs, Persians, Africans and Indians, did not erase the common thread. It diluted it, it mixed it, it layered local ancestry on top of it, but it did not break it. And the most unexpected finding was not that Jewish populations are related to each other. It was who else they are related to. Subscribe. Because the DNA of the Jewish people tells a story that will make everyone, regardless of their background, rethink what they thought they knew about ancestry, identity, and the question of who is related to whom. Modern Jewish populations are divided into 3 major groups, each shaped by where their ancestors spent the last 2,000 years. The Ashkenazi Jews settled in Central and Eastern Europe, Germany, Poland, Russia, Lithuania, Hungary. They developed Yiddish, a language blending Hebrew with medieval German. They built a cultural world of synagogues, scholarship and commerce across the cities and shtetls of Europe. Today, Ashkenazi Jews are the largest Jewish group, comprising roughly 70 to 80% of the global Jewish population. The Sephardi Jews settled in the Iberian Peninsula, Spain and Portugal. They developed Ladino, a language blending Hebrew with medieval Spanish. When the Spanish Inquisition expelled them in 1492, they scattered across North Africa, the Ottoman Empire, the Balkans, Italy and the Netherlands. Their genetic profile carries Iberian, North African and Middle Eastern signatures. The Mizrahi Jews never left the Middle East. They remained in Iraq, Iran, Yemen, Syria and Egypt. They spoke Judeo-Arabic, Judeo-Persian and other regional Jewish dialects. Of all Jewish groups, the Mizrahi retain the highest proportion of unbroken Middle Eastern ancestry because they stayed closest to the point of origin. When scientists compared the DNA of these 3 groups, they expected to find that 2,000 years of separation had pulled them apart genetically. That Ashkenazi Jews would look European, that Sephardi Jews would look Iberian, that Mizrahi Jews would look Iraqi or Yemeni. That is not what the data showed. A landmark 2010 study published in the American Journal of Human Genetics analyzed the genomes of 305 Jewish individuals from 7 major communities using high-resolution genotyping. The senior author, Harry Ostra of NYU, described the results. All Jewish populations fall into a large genetic cluster that is distinct from non-Jewish populations, with population-specific subgroups showing different levels of Middle Eastern ancestry and local admixture. Ashkenazi, Sephardi, Italian and Syrian Jews clustered more closely to each other than to the European or Middle Eastern populations they had lived among. The European Jews were genetically closer to Middle Eastern Jews than they were to their European neighbors. 2,000 years of living in Europe did not make Ashkenazi Jews European. It made them a mix. But the Middle Eastern core remained. The root held. The Ashkenazi finding was the most dramatic and the most debated. A study published in Nature Communications by the Ashkenazi Genome Consortium sequenced the complete genomes of 128 Ashkenazi individuals and compared them to non-Jewish European populations. The results revealed that modern Ashkenazi Jews trace their ancestry to roughly 350 individuals who lived approximately 600 to 800 years ago in Central Europe. 350 people. Every Ashkenazi Jew alive today, roughly 10 million people, descends from a founding population smaller than most high school graduating classes. This extreme bottleneck, known as a founder effect, explains one of the most distinctive features of Ashkenazi genetics. The limited founding population means that certain genetic variants, including disease-causing mutations, were present in the founders and amplified as the population expanded. The BRCA1 and BRCA2 mutations, which dramatically increase breast and ovarian cancer risk, are carried by roughly 1 in 40 Ashkenazi Jews compared to roughly 1 in 250 in the general population. Tay-Sachs disease, Gotcha disease, and familial dysautonomia are all disproportionately common in Ashkenazi populations for the same reason. The mutations were in the 350 founders. The population expanded. The mutations expanded with it. But the bottleneck also revealed something unexpected about ancestry. By analyzing the genomes of the 128 individuals, the consortium determined that Ashkenazi ancestry is roughly an even mix of Middle Eastern and European origins. The paternal lineages, traced through Y chromosome DNA, are predominantly Middle Eastern. The haplogroups J1 and J2, which are associated with the ancient Levant, appear at high frequencies. The maternal lineages, traced through mitochondrial DNA, show a more complex picture, with a significant proportion tracing to European women. The interpretation is straightforward and supported by the data. Jewish men from the Levant migrated to Europe roughly 2,000 years ago. They married local European women. Their descendants maintained Jewish identity, religion, and community, while carrying a genome that was half Middle Eastern and half European. If you have made it this far, subscribe. The next chapter reveals the finding that made headlines around the world because it answered a question that is not just scientific but deeply political. The children were Jewish. The grandchildren were Jewish. The great-grandchildren were Jewish. For 2,000 years, the community maintained its identity, its religious practice, and its genetic coherence, even as European DNA was incorporated through the maternal line. The culture held the genetics together, and the genetics 2,000 years later still show the seam between the Middle Eastern fathers and the European mothers. Here is the finding that made the research politically explosive. When scientists compared Jewish DNA to non-Jewish populations from the Middle East, the closest genetic match was not a distant or abstract connection. Jewish populations, particularly Mizrahi and Sephardi Jews, cluster genetically with Palestinians, Druze, and Bedouin populations of the Levant. The shared Y chromosome haplogroups J1 and J2 appear at high frequencies in both Jewish and Palestinian populations. The autosomal DNA, the total genome from both parents, shows significant overlap. The genetic distance between a Mizrahi Jew from Iraq and a Palestinian from the West Bank is smaller than the genetic distance between either of them and a Northern European. The DNA says they are cousins, not metaphorical cousins, genetic cousins. Descended from the same ancestral population that lived in the Levant roughly 3,000 to 4,000 years ago, before the historical events that separated them into different religious, cultural, and national identities. The 2010 Ostra study confirmed this explicitly. Jewish populations from Iran and Iraq clustered closer to non-Jewish Palestinians, Druze, and Bedouin than to European populations. The Middle Eastern root of Jewish ancestry is shared with the non-Jewish populations that remained in the region. This finding does not resolve any political dispute. DNA does not determine borders or rights, but it establishes a biological fact that complicates every narrative that frames the conflict as one between indigenous and foreign populations. The genetics say both populations are indigenous to the same land. The separation is historical and cultural. The biology is shared. The Khazar theory, which proposed that Ashkenazi Jews descended from converted Turkic Khazars from Central Asia rather than from the ancient Israelites, was directly addressed by this research. Ostra stated plainly, there is no evidence for the Khazar theory. The genetic data shows clear Middle Eastern origin for the paternal lineages of Ashkenazi Jews, consistent with Levantine ancestry. The Khazar hypothesis, which had been used politically to challenge Jewish claims to Middle Eastern origin, was not supported by any genomic evidence. The DNA did not pick a side. It established a fact. Jewish and Palestinian populations share deep common ancestry from the ancient Levant. The genetic distance between them is smaller than either would expect, and the shared haplogroups that connect them are 3,000 years old. The Ashkenazi, Sephardi, and Mizrahi represent the largest branches, but the Jewish diaspora produced smaller communities that traveled further, stayed longer, and developed genetic profiles that surprised even the researchers who studied them. The Beta Israel, the Jewish community of Ethiopia, had practiced Judaism for centuries in the highlands of East Africa. When their DNA was analyzed, the results showed a predominantly East African genetic profile with a small but detectable Middle Eastern component. The Middle Eastern signal was modest compared to Ashkenazi or Mizrahi populations, suggesting either a small founding group of Middle Eastern migrants who married into the local population, or a very ancient connection that has been diluted over centuries of isolation. The Beta Israel present a challenge to simple genetic definitions of Jewish identity. Their religious practice is authentically Jewish. Their genetic profile is predominantly East African. The DNA and the identity do not align in the way they do for other Jewish populations. Israel recognized the Beta Israel as Jews and facilitated their immigration in massive airlifts in 1984 and 1991. The decision was made on religious and cultural grounds, not genetic ones. The Cochin Jews and Beni Israel of India represent another diaspora extreme. The Cochin Jews of Kerala claim a presence in India dating back 2,000 years, Their DNA shows Indian and Middle Eastern admixture in proportions that confirm a migration from the Levant, followed by centuries of intermarriage with local Indian populations. The Beni Israel of Maharashtra show a similar pattern, with Y chromosome lineages tracing to the Middle East and autosomal DNA showing significant Indian ancestry. And then there is the Cohen modal haplotype. Among Jewish men who identify as Kohanim, members of the priestly lineage descended from Aaron, the brother of Moses, a specific Y chromosome signature appears at dramatically elevated frequencies. The haplogroup, a subset of J1 and J2, has been estimated to trace back roughly 3,000 years, consistent with the biblical timeline of the priesthood. The priestly lineage is passed from father to son, the Y chromosome is passed from father to son, and the Y chromosome pattern found in modern Kohanim is consistent with descent from a common male ancestor roughly 3,000 years ago. The DNA did not prove the biblical Aaron existed, but it proved that the men who claim descent from a single male ancestor 3,000 years ago do, in fact, share a Y chromosome signature consistent with that claim. A genetic signature passed from father to son for 3,000 years. Still detectable, still consistent, in men who have been identifying themselves as priests since before the Romans destroyed the temple. The oral tradition said they were related. The DNA confirmed it across continents, across millennia. The thread held. So here is what happened when scientists compared Jewish DNA to every other population on Earth. They found that Jewish communities scattered across 4 continents for 2,000 years still share a genetic core traceable to the ancient Levant. The Diaspora mixed the DNA but did not erase the origin. The root survived in every branch. They found that 10 million Ashkenazi Jews alive today descend from roughly 350 founders who lived 700 years ago. That bottleneck concentrated both brilliance and disease in a single population, producing the highest rate of Nobel laureates per capita and the highest rate of BRCA mutations in the world from the same small founding group. They found that Jewish and Palestinian populations are genetic cousins, sharing Y chromosome haplogroups, sharing autosomal ancestry. Descended from the same ancient Levantine population separated by history and religion, connected by biology. They found the Khazar theory unsupported by any genomic evidence. The Ashkenazi are not converted Turkic nomads. They are Middle Eastern men who married European women 2,000 years ago and maintained their identity through every century since. They found Jewish communities in Ethiopia, India, and Central Asia that carried the Middle Eastern thread in varying concentrations, proving that the diaspora reached further and lasted longer than most historical records document. And they found a Y chromosome signature in the priestly lineage that is consistent with descent from a single common ancestor roughly 3,000 years ago. A genetic thread connecting modern Kohanim to a founding figure at the beginning of the biblical priesthood. The results shocked everyone because they confirmed something that was simultaneously expected and impossible. After 2,000 years of exile, persecution, conversion, intermarriage, and dispersion across the entire planet, the genetic thread connecting Jewish populations to each other and to the ancient Levant did not break. It frayed, it thinned, it mixed with every population it passed through, but it did not break, and when scientists finally had the technology to look for it, there it was, in the chromosomes, in the haplogroups, in the shared mutations. In a priestly lineage that has been passing itself from father to son since before most civilizations on Earth existed, the thread held for 3,000 years, across every continent, through every exile. That is what the DNA says, and the DNA does not negotiate. 350 founders for 10 million people. A 3,000-year-old priestly Y chromosome genetic cousins with Palestinians, Jewish communities from Ethiopia to India, carrying the Middle Eastern thread in different concentrations, and a diaspora that scattered a people across 4 continents for 2,000 years, without breaking the genetic connection to where they started. The scientists compared Jewish DNA to every population on Earth and found something that no other diaspora population has demonstrated at this scale. Genetic coherence across millennia, maintained not by isolation but by identity. Drop a comment. Did any of these findings change what you thought you knew? Which one surprised you the most? Subscribe for more. We will see you next time.